Welcome to the lecture. My name is Ivan Somongo from the School of Bio and Applied Sciences, Department of Biological Sciences. So today's lecture will be on the unit BBY1103, Cell Biology and Genetics. So today's topics we're going to cover are 4 topics. We're going to cover Introduction to Cell Biology, cell structure and function, cell membrane, and also look at cell division. Let's begin by defining what is cell biology. Now, cell biology is a branch of science. Sometimes it's also known as cytology, where we study the structure, functions, and behavior of cells. We all know that cells are the basic unit of life. This is knowledge taught to us even at primary school level, and that all living organisms are made up of cells. And it's the same way like the way matter, the basic unit of matter is atoms. In living organisms, the basic unit are cells. Now, cells appear as single cell. We have certain organisms that are only one cell. Then we have certain organisms which are made up of many cells, which are known as multicellular organisms. The single-celled organisms are things like the protozoa, or the bacteria. Most of the time we like calling them microorganisms. And then the multicellular organisms are your organisms like plants, animals, fungi, which are made up of many, many cells joined together. So cells vary in size and structure, but that variation is basically for their specialization, because different cells perform different functions. Therefore, they have to specialize for their particular function. That's why we have different organisms. That's why we have different tissues. We have different organs which perform different things. For example, the work the red blood cells do is different from what the white blood cells do. I'm sure that is knowledge we already are aware of. However much different the cells are, they all share some basic structural components. And that is what we are going to look at in today's class. Now, before we do that, we could look at what the history of cell biology. Cell biology, we can say, started way back in 500 BC, but back then, what was being reported was just the morphological characteristics of tissues that the people were observing when they were doing autopsy on dead or cadavers, dead organisms or cadavers. And they were able to now report this, what they were seeing, and that could be said could be the basis of cell biology. However, cell biology began proper around 1590 with the discovery of the microscope. The microscope is an instrument that enables us to magnify images, and therefore we are able to use it to observe cells, because cells are microscopic. They are very small. They cannot be observed with the naked eye. Therefore, with the discovery of the microscope, we were now able to begin to see into the tissues and see the particular cells. So the microscope was discovered by people who are not actually scientists. They were spectacle makers, meaning they were making spectacles for the people at that time. And they experimented, because they were working with lenses, they experimented in coming up with a higher magnification of objects. So the two, Hans and Zacharias Janssen, were accredited with discovering the microscope. Actually, Hans is the father to Zacharias, and Zacharias is credited more in the discovery or that innovation of coming up with a microscope. But around there's still more controversy, because other sources also say there are other people who contributed to the microscope, like a fellow spectacle maker in the area where Hans and Zacharias lived. He also contributed to making of the microscope. There are other stories that there was a traveler who came with the knowledge, and the two just picked up that knowledge to develop the microscope. However, in 1658 is where we see the microscope beginning to be used now to examine structures. The scientist Jean Swammerdam was the first one to begin to report the observation of microscopic structures in insects, and he was also able to report about the red blood cells using the microscope. Again, in 1665, we have another scientist, an Englishman called Robert Hooke, who decided to observe cork. Cork is like dead wood, and he was able to see through a microscope that he made by himself that the cork had certain holes, which he called cells. In fact, that is the first time we begin to use the word cells to describe these structures. So Robert Hooke was able to see those cells, but actually, interestingly, what he was looking at were not cells, but the spaces that were left by the cells, because the cork is actually dead wood, and the regions he was looking at are actually the spaces that the cells were occupying, which were now already dead and they were not there. But he described that as cells, and that's where the idea of cells now came about. In fact, his discovery was not appreciated for another 200 years, because between the 1600s to the 1800s, in between, we don't have much reports about cells and cell biology. However, around 1667, we have the scientist known as Leeuwenhoek, who was studying microorganisms using the microscope again, and he was able to see small organisms like protozoa, and also 9 years later, he was able to report about the bacteria. Later, we have another Englishman in 1833, now that is 200 years later, who now began to observe cells. He was called Brown. He began to observe cells and noticed that in the cells, at the central part of the cell, he noticed a structure always appeared at that central part, which we now call the nucleus. Other scientists also now contributed to the whole process. Between 1835 to 1839, we have Dujardin and Pukinye, who are now beginning to explore not the external part of organisms, not the entire cell structure, but now looking internally inside the cell. What are the components that are found inside the cells? And you can find many reports about some of their findings, some of the organelles or some of the tissues within biology. Some are actually named their names, or the name Pukinye is a common name. Then in 1858, now what we know as the cell theory began to be formulated. Now, this is a theory that explains about cells. So we are going to look at what it actually means. And its contribution was done by two scientists known as Sheldon and Schwann, who were working on different cells, and they were able to not formulate the cell theory, but they gave the idea about the cell theory. Now, Sheldon and Schwann were two scientists. They were working in the same institution. One was a botanist, the other one a zoologist. Therefore, they worked in the same institution, but not in the same lab. And in 1839, it is said that they had lunch together. In the process of talking, they discovered that the cells they were looking at from plants and animals were almost similar. And that the idea now came to them that probably different organisms are made up of the same cells, or cells which have more or less some common structures or some similarities. Now, in 1852, a Polish scientist working in Germany known as Riemek also began to study cells. He was a pathologist, and he noticed that cells were normally generated from other cells, meaning a cell would divide and create a new cell. And he was able to publish that information. And then in 1858, another Polish scientist, again interestingly, the two were working in the same area. So the Polish scientist known as Vicho wrote an editorial essay talking about how cells rise and divide. It is known as it was titled "All Cells Arise from Cells." And this editorial, or this essay that he wrote, publicized or popularized the cell theory. Actually, an editorial essay that he wrote was not his work. He didn't go to the lab and do the work himself. He compiled information from other scientists who had worked on cells and was able to come up with that essay. And most of the work he reported were work done by Riemek, because Riemek also studied the same thing about cell division. But he did not credit Rimek with that knowledge in his essay. So we are told that Virchow is the one who came up with the concept of cell theory, but actually other scientists like Sheldon, Schwann, and Rimek are the ones who now compiled who came up with the information that he was now able to use as to come up with the essay. But it is his essay that now popularized the idea that cells have certain particular qualities, which we call the cell theory. In fact, there's still controversy as to who came up with the cell theory. Was it Virchow, Rimek, or should it be credited to Schleden and Schwann? So what is the cell theory? The tenets of the cell theory state that, and you know a theory is something that is there until it is proven otherwise. So for now, we know that all living organisms are composed of cells. That is, they're all made of cells. There are no living organisms which are made of other things apart from cells. Then here probably someone would argue with me and say that talk of viruses, but that would be another topic where we say viruses you can say they are living. Some people say they are not living. That is a topic for another day. So all living organisms are composed of cells, and cell is the basic structural and functional unit of life, meaning all organisms, the basic structural and functional unit is the cell. Then the third tenet is that all cells are created by other cells, meaning they arise from pre-existing cells. So those are the three tenets that guide the cell theory and that we use when we are talking about cell biology. So let us now turn to the cell structure. We've done the introduction. Let us turn to the cell structure. So what is a cell? A cell is a small membrane enclosed unit that is filled with a fluid. It's not necessarily a fluid, but you can say it's a gel, and which in that fluid there are floating particles. And we're going to see some of the particles are organelles, some of the particles are enzymes, some of the particles are metabolites. These are found within that region, this aqueous solution within that region. And then we say that cells are infinitely varied, meaning there are many different types of cells. But however, internally, the cells are almost similar when you look at them internally, meaning most of them are composed by the same molecules. When you talk of carbohydrates, proteins, lipids, the same molecules are found in every cell. You pick a bacterial cell, an animal cell, a plant cell, you'll find the same types of components. They might not be exactly in the same structure, but they are the same components. Then they participate in the same chemical reactions. If you look at the different cells, you'll see the same chemical reactions happening inside them. They have the same genetic material in form of DNA with the same structure. They might vary in their order, but the same basic structure is there. Look at it from this point that we have buildings. Okay, buildings are of different designs, but most of them, what you can say the stone buildings, are all made of stones. But the stones are arranged in different packages to get different types of buildings. So the same way we have DNA, which makes a genetic information, but it's packaged differently in different cells. That's why we have varied organisms. And interestingly, the DNA, the genetic information is written in the same code. So the same code is used everywhere. So you can say the same language is used to translate the genetic information into the different organisms that we have. Now, like I said, those cells are varied. They share certain structural features, meaning the three main components you'll find in a cell is there'll be an outer boundary, and that one we call it the cell membrane. Then inside the cell, you'll always find a centrally located structure which we call the nucleus, and in certain cells we call it the nucleoid. We're going to look at the difference. And then there is the aqueous part, which I talked about earlier. That region is known as the cytoplasm, which we said is a mixture of the fluid part and some particles. So the fluid part is what we call the cytosol. And the other parts which fill the interior Different suspended particles, which I had mentioned earlier, are things like organelles, enzymes, metabolites, etc. Those ones are found suspended inside the cytoplasm. Previously, we classified cells as animal cells, plant cells, bacterial cells. But in science, we like to classify classify cells in terms of two classifications. And the classifications is based on whether the cell contains a membrane around the nucleus. So as I had said earlier, the nucleus is a structure that you find mostly centrally in the cell. Now, cells that have a boundary around the nucleus are known as eukaryotic cells. These cells also have other organelles which also have a boundary. And examples are plant and animal cells. Then we have a second class of cells which we call the prokaryotic cells. These are cells which do not have a membrane or a boundary around their nucleus. So you can say their DNA is floating at a certain region, but there's no boundaries separating it from the cytoplasm. And examples are things like bacteria. And prokaryotic cells are further classified into two groups. We have eubacteria and archaea. Now, eubacteria are your everyday bacteria, the ones we spoil our food, cause diseases, and such kind of things. But archaea are bacteria, or archaea are bacteria or prokaryotics. You can say prokaryotic cells that live in very harsh conditions. These are like the bottom of the sea where the oxygen is lacking, in hot springs where we have very high temperature, in things like the cow's stomach where maybe it is acidic, things like in concentrated brine, which is very salty water, which you find these prokaryotic cells. Now this is interesting because cells normally survive under certain conditions. And these conditions are considered harsh because something like high temperature will cause denaturation of things like proteins within the cell, and it will cause the death of the cell. Things like lack of oxygen, we know oxygen is required by cells to perform certain functions like cellular respiration so that it can make energy for the cell. Therefore, surviving in a place where there is no oxygen is quite a harsh condition, but we have certain prokaryotics which survive in that condition. What they simply do, they have alternative things, or alternative, let's say it's oxygen, they have an alternative for that oxygen which they use to generate energy for the cell. Some live in concentrated brine, which is salty water. And you know, whenever you put a cell in salty water, it will shrivel because water will be attracted out of the cell into the brine solution and then cause the cell to die. But these particular cells are able to survive under those conditions. So they are classified differently. The two, eubacteria and archaea, are actually very distinct if you look at them in terms of their biological or their biochemical action or activities. So let us look at one cell, the prokaryotic cells. Here I've included a diagram showing a prokaryotic cell. As you can see, it is covered by a membrane around it, but the nucleus here, it's not a nucleus, but it's what we call a nucleoid, and the DNA is just floating around here. We would have expected to see a boundary here, but there is no boundary. This one has other structures, flagellum, which we are going to look at. And they're very small. I don't know whether the camera can capture this. This is 1 micrometer, meaning it is a very, very, very small structure. 1 micrometer is like 1,000 millimeters. 1,000 micrometers make 1 millimeter. So that is how small this is. So what are some of the properties of prokaryotic cells? One is that they are spherical, rod-like, or corkscrew shaped. They are small, like I had stated, very, very small. They often have a tough protective cell wall. As you can see, there is a cell wall which is covering the cell. And then, like we had said earlier, they lack membrane-bound organelles. So within here, you would find different organelles, as we're going to see with the eukaryotic cells, but in prokaryotic cells, these organelles are not bound around membrane. Here, you have indicated that this one has ribosomes floating in the cytosol. You can't see a clear boundary separating them from the cytosol. The other type of cells are eukaryotic cells. Eukaryotic cells are larger and more complex than prokaryotic cells. Like we said, they have a membrane around their nucleus, and they have many organelles which also have membranes. And these organelles, the term organelles simply is a projection of organs, small organs. So organelles would mean small organs. And these small organs perform different functions for the cell. They are like different areas that contribute towards the cell. And the presence of certain organelles helps in specialization of the cell. If a cell is to produce more proteins, then it would have the organelle that is producing the proteins in higher numbers than other organelles. Then eukaryotic cells, which this is also different with prokaryotic cells. Eukaryotic cells have what we call a cytoskeleton, like a skeleton structure around the cell. This is made of microfilaments and microtubules. These are string-like structures around the cell, which now help in giving the cell shape and also allowing intracellular movement within the cell. This intracellular movement, try to figure it like the muscles that we have in the esophagus or in the throat region. They help to push down food. So the movement of the muscles move the food in our throat area. So within the cell, also the movement of these muscles would help, not muscles, but these filaments and microtubules would help the movement of certain substances inside the cell. So some cells live as single-celled organisms, we had mentioned earlier, or some eukaryotics live as single-cell organisms, such as amoeba and yeast. Others are multicellular, meaning they're made up of many cells, such as plants, animals, and fungi. So let us look at some differences between prokaryotic and eukaryotic cells. First, we said prokaryotic cells, one difference is prokaryotic cells are small, while eukaryotic cells are larger. These ones are more than 10 micrometers, while prokaryotic cells are about 5 micrometers and less. Prokaryotic cells, most of the time you'll find that they are unicellular, meaning one cell creating an entire organism, but in eukaryotic cells are found in organisms which are multicellular. Like we said, prokaryotic cells do not have a membrane around the nucleus, and they also lack organelles that are membrane bound. While eukaryotic cells, they have a nucleus which is membrane bound, and also they have many organelles which are also membrane bound. The DNA that in prokaryotic cells is circular, meaning DNA is usually like a string. This one is circular. There is no beginning end to the other end. It is continuous. While in eukaryotic cells, the DNA is linear, where it is also combined with proteins known as histones, to form the structure we call the chromatin. In prokaryotic cells, we do not have the proteins attached to the DNA. In prokaryotic cells, again, they have the ribosomes, which are very small. If you remember the structure or the drawing of the prokaryotic cell, the bacteria, you will see there are ribosomes. The ribosomes are small. In eukaryotic cells, the ribosomes are large. In fact, they are compared in terms of their sedimentation coefficient. But that means if you centrifuge them, how will they sediment? And that's why you have the term there 70S and 80S. These are what you call Svedberg units. So this tells you the ribosomes could be lighter than the ribosomes in prokaryotes would be lighter than the ribosomes in the eukaryotes. Another thing we already mentioned is that the prokaryotic cell has no cytoskeleton, while the eukaryotic cell has a cytoskeleton. Another feature is prokaryotic cells, the structure they use for moving, which we call the flagellum, is normally rigid. But in eukaryotic cells, the structure for mobility is flexible, and they have both flagella, which is a single tail-like structure, and cilia, which are hair-like structures which could appear on the side. The structure is also made of different types of protein. This one is made from flagellin, and in eukaryotic cells is made from tubulin. Those are different types of proteins. In prokaryotic cells, cell division is by binary fission. The cell just divides into two. But in eukaryotic cells, cell division is by mitosis or meiosis. We are going to see what we mean by those two. But these are elaborate cell division processes which are very accurate to try and ensure that the DNA being transferred from one cell to the new cells the number is accurate. So we are going to see how that process happens and how the division is very accurate, which we call mitosis and meiosis. Prokaryotic cells, most of the time reproduction is asexual, while in eukaryotic cells reproduction can be asexual or sexual. You remember sexual reproduction is when two cells come together to form a zygote. In asexual reproduction, you don't need not that you don't need, but you do not have two cells coming together. It is one cell dividing into two, or an organism just being propagated from one particular cell. Is what we call asexual reproduction. And then in prokaryotic cells, we have a huge variety of metabolic pathways, while in eukaryotic cells, they have common metabolic pathways. Now, metabolic pathways are processes in which these cells are able to convert molecules or compounds from one form to the other. They are catalyzed by enzymes, and if you study them, you'll find that they are made up of a number of steps where the metabolite or the compound or the molecule is converted from one form to another form to another form to the final form in which the cell would utilize. Now, in prokaryotic cells, they have varied ways of getting to the final point. Look at it in the point that if you wanted to go to, let's say, a town, you could go through the main highway, or you could go through certain footpath routes to the town. So at the end, you'll always get to the town. But prokaryotic cells are the examples of where you use the footpath. The footpath, you can use different footpaths to get to the final destination. But in eukaryotic cells, you use the highway because it is one way of doing things. So this variety in prokaryotic cells is what also gives us the variety of bacteria, which can live in different conditions because they can use different processes to use different substances to get to the products. But eukaryotic cells are limited to which pathway they use to get to their final products. So you could read more about metabolic pathways to understand what we mean by huge variety of metabolic pathways and common metabolic pathways. So that's a diagram of a eukaryotic cell. As you can see, it has various organelles. First of all, on the outer side, it has a plasma membrane, which we call the plasmalemma. It is a boundary around the cell. Then we have the nucleus, which we talked about, covered by a nuclear membrane. As you can see, the nuclear membrane is not continuous. There are some spaces here which we call the nuclear pore. This is where now substances can go in and out of the nucleus. The nucleus, there's an area called the nucleolus. Then there is chromatin in the nucleus. These are DNA which are found within the nucleus. There are other organelles like the rough endoplasmic reticulum, peroxisome. We have a chloroplast there. So this would be a plant cell. but not a very good one, but it's just trying to show the different types of eukaryotic cells, eukaryotic cell organelles, Golgi body, mitochondria, ribosomes free. There are ribosomes that are free on the cell, then there are ribosomes that are attached to the rough endoplasmic reticulum. There is a smooth endoplasmic reticulum. We have a lysosome, and you have what we call the vesicles or vacuoles. So let us look at these various organelles and try to see what are their functions in making this entire cell do what it normally does, you know, keeping the person or the organism alive. So we start with the cell wall. Eukaryotic cells do not have a cell wall, but this is some general organelles which are put together so that we can study the different organelles that we normally encounter within a cell. Cell wall is it's more of a rigid structure. It is found in bacteria and plant cells. So animal cells do not have the cell wall. Plant cells, although they are eukaryotic cells, they also have a cell wall. They are made up of microfibrils, you know, which are like fibers, which are made from different materials in the different organisms. Like in bacteria, it is made using peptidoglycan, which is, I think, would be a protein mixed with a carbohydrate. We have in plants, we'd have cellulose, which is a carbohydrate, making up the cell wall in plants. And also in certain plants, you'd have other compounds such as pectin and lignin. These are mostly to harden the cell wall. You notice that there are certain plants which are kind of soft. There are certain plants like trees which have certain regions which are very rigid and very hard. Now this is a play on the various substances here to be able to make those different kind of structures. Now the cell walls contribute to the rigid structure of those particular organisms. Now, like I said, the cell wall possesses some breaks or some spaces within them. This space allows for certain strands, known as the plasmodesmata, to travel from one cytoplasm to the other, so that this cytoplasm of cells that are close together can be able to communicate. So that helps the cells now to share information and also to share resources as the need be. So what is the function of the cell wall? First, it's to protect the delicate inner parts of the cell. So like I said, it's a rigid, strong structure which helps the cell to protect its inner delicate parts. It provides the rigid shape of the cell. Remember, since things like bacteria do not have a cytoskeleton, they would require an outer wall that is strong enough to ensure that the cell retains its shape so that things inside are not squeezed or something like that. Then it also, like I had said, allows passage of substances in and out of the cell. This is very important because the cell needs to be nourished. The cell needs to remove waste substances. Therefore, that wall should be able to allow substances to move in and out of that particular cell. Another organelle, which is now after the cell wall in certain organisms, but in eukaryotic cells mostly it is the one that forms the outer boundary, is what we call the cell membrane. In other terms, we call it the plasma membrane. In certain terms, we call it the plasma lemma. So this one covers the outer part of the cell. If you look at the diagram I was projecting earlier, you could see the cell membrane outside the cell. Now it is composed of lipids and proteins which form a thin, pliable, hydrophobic barrier. So what do we mean by a thin, pliable, hydrophobic barrier? We say it is pliable, meaning it can be moved around. Okay, it is not a rigid structure. This is important because the cell may want to change shape here and there, as we're going to see in processes like phagocytosis, where the cell wall has to be able to move around, to be fluid, so that it can be able to accommodate certain substances moving into the cell and out. So it is pliable, it can move around. We also say it is hydrophobic, meaning it repels water. Okay, we are going to look at later what that particularly means. And then it is a barrier. A barrier means it blocks certain substances from moving into the cell. But we normally say it is a semi-permeable barrier or semi-permeable membrane, meaning it allows certain substances to go in and out of the cell, but it blocks certain substances from going in and out of the cell. So that is important because you want the cell to take in what is important to it and leave out what is not important to it or what can cause harm to the cell. So what are the functions of the cell membrane? Like I'd already mentioned, it forms a barrier to allow free passage to a barrier. Sorry, it's a barrier, and that means it blocks the free passage of inorganic ions and most other charged or polar compounds. So it blocks them from getting in, but it allows some of them through what we call the transport proteins. We're going to see what are these transport proteins that allow passage of certain ions and molecules through the cell membrane. These ones, things like sodium ions, potassium ions, are important for the functions of the cell, so they are allowed through, but they are carried through in what we call transport proteins. Within the cell membrane also, you'll also find what we call receptor proteins. Now these are proteins that are on the surface of the cell membrane that act as receptors. They gather information which they send into the cell. So they sense information from the external environment and then transfer that information into the cell so that now the cell can act or react to that environmental stimuli. So those ones are on the surface. So the membrane, apart from being a barrier, it also acts as a sensor. Okay, it's the way the cell can sense what is happening in the environment. Then some membranes also have enzymes which now participate in certain biochemical reactions that the cell participates in. And also importantly, we said the cell membrane is pliable, meaning that it allows the cell to change its shape and its size. Sometimes a cell needs to divide, therefore it needs to change its shape. Sometimes it has to grow, it will increase in size for it to grow. So the membrane has to be pliable to allow for this different change of shape to happen. Another organelle we find in the cell is the nucleus. The nucleus is enclosed in a double layered membrane. So even the cell membrane is double layered. The nucleus also has a double layered membrane. I think what I forgot to mention at the beginning is most of these membranes that surround the various organelles and the cell itself are double layered. We're going to see about membrane and you'll see what that double layer means. So we normally call it the nuclear envelope. And like I had mentioned earlier, that envelope or that membrane contains pores. Okay, pores are like holes. Now these pores, they allow passage of large molecules in and out of the nucleus. Things like DNA. When DNA has been synthesized in the nucleus, or sorry, not DNA, but RNA, when RNA or ribonucleic acid has been synthesized from DNA inside the nucleus, it has to travel out of the nucleus to the cytoplasm to be used by other organelles like the ribosome to create proteins. So that RNA has to find a way of traveling out of the nucleus into the cytoplasm. So it does that by moving through the pores, plus other substances like DNA is constantly being recreated. So the components of the DNA to recreate more DNA will have to come from outside the cell through the various nourishments, get into the cytoplasm and find its way into the nucleus so that they can be used to make new to repair parts of the or when you're having cell division to create new DNA so that you can have the cell division to happen with equal amounts of DNA moving to the different cells that are being created. So the envelope also covers the region inside the nucleus we call the nucleoplasm. That is where you'll find the DNA floating in form of chromosomes. I'm sure you can read more about chromosomes to understand the relationship between chromosomes and DNA. But it's basically that DNA makes up things known as genes, and genes together form chromosomes. So what are the functions of the nucleus? Is to coordinate the activities of the cell and also coordinate the activities of the cell organelles. It is the nucleus that now picks up the information, especially on a cell that has to produce certain substances, and it now directs the various organelles to produce those particular substances. It also functions in repair work. The cell is constantly repairing different structures here and there, and therefore it would require to be coordinated so that it doesn't get out of hand. It coordinates also cell division and gamete production. The process of cell division is when a cell divides to create new cells. And gamete production is when we are producing cells that, things like sperm cells and egg cells or ova, which have half the amount of DNA as an original or what you call a somatic or body cell. So to create those gamete cells, it is the nucleus that coordinates all that particular work. Another organelle is the mitochondria. Mitochondria also, like I've said, most of these organelles would have a double membrane. The mitochondria also has a double membrane, and the inner membrane, because there are two, the inner membrane is normally folded into structures we call the cristae. And the reason why they are folded, it is on this inner membrane is where we have certain chemical reactions known as cellular respiration. It happens in that inner membrane. So the folding would be to increase the size so that more action can happen within that particular membrane. So on that membrane there are various proteins that participate in this what we call cellular respiration, which is simply an energy transduction kind of a reaction where through the oxidation of food, energy can be transferred from food, which nourishes the cell, and stored in form of chemical energy known as ATP. Now this ATP can then be used by the cell to power other reactions. So that energy is harnessed or harvested from food by oxidation. So those reactions happen within the inner membrane of the mitochondria, and that's why it is folded, and we have those structures known as cristae. Interestingly, mitochondria, compared to other organelles, is the other organelle apart from the nucleus that also contains some DNA. And it is also able to reproduce by dividing. So our nucleus, our mitochondria can divide into two and create more mitochondria controlled by its internal system. So we are normally told that mitochondria could have been bacteria that were swallowed by a eukaryotic cell, and that is why they would have their own DNA and also be able to divide on their own. These now are processes where we are trying to explain the evolution of eukaryotic cells. You'll find this information that they could have been derived from bacteria which were swallowed by a eukaryotic cell. So next we have another organelle called the chloroplast. Chloroplast is only found in plants and certain algae. We have heard of the green algae. It is a green organelle and it is large. Okay, the structure is large. It also possesses a double membrane, and inside the chloroplast would be the fluid structure or substance we call the stoma. And floating within the stoma are interconnected stacks, okay, membranes which are like stacked together, known as grana. Now this grana contain the green pigment known as chlorophyll, which participates in photosynthesis. And just like the mitochondria, the chloroplast is the one that generates energy for the plant or the algae. What it does, it conducts the process of photosynthesis. In photosynthesis, we use light energy to be able to combine carbon dioxide and water to create carbohydrates. Therefore, the energy that is coming from the light is captured in this product known as the carbohydrate. Now, this carbohydrate can later now be used to create ATP energy, or can also be used to create energy in other ways for the particular plant. So the chloroplast does the work of capturing that light energy and storing it in a chemical energy form for the plant to be able to use. I think this is why they say plants make their own food through the process of photosynthesis. They also contain their own DNA and also reproduce by dividing. And again, they're also believed to have come about or evolved from bacteria, but this time photosynthetic bacteria that were engulfed by a eukaryotic cell. That's why they have their own DNA and also they can reproduce by dividing. Then another organelle we have is the endoplasmic reticulum. This is an irregular network of interconnected spaces which are enclosed within a membrane. Their work is mostly to export materials that have been synthesized inside the different organelles. If you remember the structure about the cell, you would see the endoplasmic reticulum ranging from around the nucleus, the nuclear membrane, stretching all the way to the plasma membrane. Therefore, through that they provide a route through which components within the cell that have been made within the cell can find a way of being exported out of the cell. You find that cells which are prominent in secretion, producing substances, like a cell that would be producing a hormone, for example, or be producing enzymes externally, or extracellular enzymes, would have a prominent endoplasmic reticulum so that it could be able to channel those substances out of the cell as quickly as possible. So we have two types of rough endoplasmic reticulum. Endoplasmic reticulum. There is rough endoplasmic reticulum and smooth endoplasmic reticulum. As I had shown you earlier, the rough endoplasmic reticulum has ribosomes attached to it, so it has kind of a rough appearance, while the smooth endoplasmic reticulum does not have ribosomes attached to it. So the work of the rough endoplasmic reticulum would also be to synthesize proteins, but actually the organelle that is involved in protein synthesis is the ribosome and not the endoplasmic reticulum. The smooth endoplasmic reticulum, like I said, it lacks ribosomes. It is involved in steroid synthesis and also in carbohydrate storage. Remember, carbohydrate is stored in organisms. We have plants storing carbohydrates in form of starch, and then we have animals storing carbohydrates in form of glycogen. So the carbohydrates are stored within the cell for purposes of providing energy to the cell when the cell would need it later, or even can help in forming structures that have been worn out and needs to be repaired. The carbohydrate can provide a source for the materials to do that. Another organelle that actually assists the endoplasmic reticulum is the Golgi body, which is a stack of flattened membranes. Their work is to package molecules which have been made in the ER for secretion or transport to other parts of the cell. So it packages substances, the various organelles, they synthesize different things. If you study about biochemistry, you'll know that different organelles are involved in different production of different substances. Like ribosomes would produce your proteins, and then another structure, like in the cytoplasm, you produce some of the carbohydrates. So to package them together to make something like a glycoprotein, for example, a substance that is made of a carbohydrate and a protein, they have to be packaged at some point. So the Golgi body would package them before they are exported out of the cell, or they are taken to another part of the cell, maybe to form a structure, maybe a membrane is broken down, so it needs a new a new membrane created for it. Therefore, the Golgi body would package that and would be used for that particular purpose to repair some worn out parts of the cell. Another organelle is the ribosome. I've mentioned it a number of times. It is involved in protein synthesis. It is a spherical organelle, and it is made up of what you call ribonucleoproteins. Now, a ribonucleoprotein is a molecule that is made up of RNA mixed with proteins. Again, if you study biochemistry, you'll know that proteins and RNA or ribonucleotides or ribonucleic acids are two different molecules. So they are combined together to make ribonucleoproteins, which are now involved in making of ribosomes. They are present free in the cytoplasm, like I had shown you in the diagram, or they may be attached to the endoplasmic reticulum. And also, as I had mentioned, they are different. In the eukaryotic cell, we have the 80S type, and in the prokaryotic cell, we have the 70S type. Another organelle is the lysosome. The lysosome is sometimes known as the suicidal bug or suicide bug of the cell, because the lysosome contains certain what you call lytic enzymes. Lytic enzymes are enzymes that break down compounds. We have enzymes that build compounds, and we have those that can break down compounds. So these enzymes that are within the lysosome are able to digest or break down the compounds of the cell. And that's why the lysosome is normally around a membrane, so that the membrane can keep the enzymes inside the lysosome. When the lysosome is broken open, the enzymes are released. They can digest the entire cell and kill the cell. They can also digest organelles and kill them. Hence, their function. What happens is they are able to digest or break down compounds. Therefore, they are used in nutrition of the cell. When the cell takes in compounds externally or molecules, before they are taken to the various organelles for different purposes, some initial breakdown of those molecules can happen in the lysosome. The lysosome, yes. It can also be involved in defense, like in the white blood cells. Whenever they take up germs or foreign bodies, when the white blood cell takes up the foreign bodies, they go to the lysosome, where the lysosome breaks them down. So it plays a role of defense. It also plays a role of cleaning up. We have certain materials within the cell that could be damaged, could be worn out, and therefore they need to be removed. So for them to be removed, they are taken to the lysosome and broken down into smaller components, which probably could be used to make other structures, because they are broken down to their basic components, and these components could also find use in making other structures. Like I had said also, the lysosome can be used to destroy old, injured, or even diseased cells. So when a cell is old and its time is up, then it destroys itself by just releasing the contents of the lysosome, so that the cell digests the contents of that particular cell. So it is involved in digestion of ingested food and also unwanted particles. So it's a very important organelle for that particular, for cells. We have also vesicles. Vesicles are formed from membranes. I'm going to show you a diagram to show you how vesicles are formed from membranes, but they actually are formed by pinching off of the membrane, and they are involved in transport. Okay, they help in transportation of substances from external part of the cell to the internal part of the cell, or from the internal part of the cell to the external part of the cell. What they do, they provide like a cover for the substances being transported, so that they do not mix with other parts of the cell, as I'll show you in the diagram. So we have two processes: endocytosis, which occurs when the plasma membrane tucks inwards and pinches off to form vesicles, and exocytosis is when vesicles from the inside of the cell merge with the plasma membrane and they release their contents outside the cells. Like, for example, things like hormones and signal molecules are secreted from inside the cell to the outside of the cell through a process known as exocytosis. So yes, the endoplasmic reticulum performs the transport process, but for especially large molecules, the process of endocytosis and exocytosis using vesicles now help in movement of those substances. Now this is a diagram showing endocytosis. As you can see, these are some substances. So sorry, the diagram is showing exocytosis as and endocytosis. Now these are some substances which are outside the cell. This is the cell membrane going round. This is the cell nucleus. So these substances, for them to enter the cell, they do not float through the membrane. What happens is they attach or come to the surface of the membrane, and the membrane folds and then pinches off to create a vesicle. This is now what we call a vesicle. Inside the vesicle, you can see there are the substances which have been picked from the outside. Then the vesicle merges with an organelle inside the cell. When it merges with the organelle, it distributes the substances into the organelle. Therefore, these substances are able to transfer or be transported into the organelle without encountering other substances or other organelles that are within that particular cell. So, like I said, the vesicle now provides a covering for those particular substances so that they do not mix with the other parts of the cell. The opposite happens with exocytosis, where the organelle pinches off. Again, it is a membrane surrounding the substances. They travel towards the plasma membrane or the cell membrane. They merge with it, and then it opens up and the contents are spilled out of the cell. So that is how vesicles help in transportation of these substances in and out of the cell. Now, when we talked about vesicles in the diagram that I had shown earlier, we also indicated stroke vacuoles. So I would like you to read more on vacuoles. We have what we call contractile vacuoles and food vacuoles, which more or less play this kind of role within certain types of cells. Lastly, we have the cellular organelle known as the cytosol. Now, cytosol is not necessarily an organelle, but it is the gel-like part of the cytoplasm. This is the part where the other organelles and other substances float within the cytoplasm. That is outside the nucleus and inside the plasma membrane. It contains organelles and a host of molecules, like I mentioned earlier. It is a site for chemical reactions, including the early breakdown of nutrients, so that the nutrients can be nutrients when they're taken in, most of the time they're in a complex form, and they have to be broken down into simpler forms there so that the various organelles can now begin to utilize those particular components that the nutrient has brought into the cell. So in the cytosol, some early breakdown of the nutrients happens. Now, the cytosol is where you'll find the cytoskeleton, which is a crisscross of filaments. I had mentioned this earlier, composed of these filaments. There are three types of filaments. There are the thin filaments known as actin, which are mostly responsible for contraction. Cells such as muscle cells are very prominent in their actin amount, because muscle cells, their main work is contraction and relaxation so that the muscles can provide movement to the organism. So actin are filaments that make up the cytoskeleton, and they're involved in contraction. Then we have some thick filaments known as microtubules. We are going to see in cell division, one main role that the microtubules do is to form what we call the spindle fiber, which helps in pulling apart chromosomes during cell division. So the microtubules are part of the cytoskeleton, but they help in that separation during cell division. And then we have intermediate filaments, which are not as thin as actin, nor are they as thick as the microtubules. And their main role is to strengthen the cell. So they act as the structural part of the cell. Now the cytoskeleton gives the cell strength, it controls its shape, drives and guides its movement. So when cells are moving and the shape and also the strength of the cell is made possible by the cytoskeleton, which is found in the entire cytosol region. So that is on cellular organelles. We have many types of cellular organelles, which some of them I have not mentioned here. I talked of contractile vacuole, I talked of food vacuole, we have the glysosomes, we have the perisomes, which you can also study more about to know about the different types of organelles. So let us move to something else, the cell membrane. Let us look deeper into the cell membrane, because the cell membrane is a very important component of the cell. Despite all the other organelles being important, cell membrane, nucleus, and the nucleus are some key and very important components. So just as a reminder, the cell membrane is the outer part. The organelles that we have looked at, you remember the mitochondria, I mentioned that the internal membrane, they have two membranes, and the internal membrane is folded. This is what we call the cristae. They are folded. I've talked of the transport vesicles. You can see them there. I've talked about the lysosome. It's just an organ like that. I've talked about the nucleus and the plasmic reticulum. As you can see, they are flowing from the nucleus towards the plasma membrane. You have the Golgi body there, which is now, after substances have been made in the endoplasmic reticulum, they can be packaged in the Golgi body, so on and so forth. So the membrane is this one on the outside, the plasma membrane. So what are some of the functions of the plasma membrane? So we mentioned them earlier, we could repeat them. First of all, they provide compartmentalization, meaning membranes are able to create compartments so that the cell can be able to perform different functions within different organelles. So as you can see, the lysosome there is packaged on its own, peroxisome is packaged on its own, the nucleus is packaged on its own. So here you have DNA, you have things like DNA synthesis, you have things like RNA transcription, DNA replication, you have the transport vesicle carrying substances in and out of the cell. So different activities are happening in different organelles. So that compartmentalization is created by those various membranes now breaking down the various organelles into different compartments so that different activities can happen in different compartments. Then they provide scaffold for biochemical activities. Within these particular organelles, there are again breakdown of regions which perform different biochemical reactions. So you can find within the organelle, you can have the separation. It's just like if you have a factory, not everything will not be done in one department. It will be separated into various departments so that different substances, different parts of that particular product could be made in different parts. So like one place could be mixing the substance or mixing the product, another one could be packaging the product, another one could be branding the product. So you'd have different stages. The same thing, when you have biochemical reactions, they do not happen all in one particular place. They happen in different regions. Therefore, the membrane would help in separation of those regions so that biochemical reactions can happen in different regions. A product could be converted from one form to another, or a substance, converted from one form to another, then transported to another part separated by a membrane, and then the product that was created at one place now is improved or modified in another particular section. That is what we call now the scaffolding or creating of those various sections. Like we had mentioned earlier, the cell membrane would also provide selective barrier, meaning it would allow certain substances in and block certain substances from going in. Cell membrane also participate in transporting metabolites. Remember, we talked about the transport vesicles, and also we talked about transport proteins. We are going to see in the structure of the membrane, there are certain channels that allow substances to be transported in and out of the membrane. So it also acts as a transport route for particular substances that the cell really need. Metabolites are substances the cell uses for it's like its nutrients. Then we said also it has receptors, therefore it acts as a way that the cell can respond to external stimuli. So the membrane around can help the cell respond to external stimuli through the receptors that it has. It also allows for intercellular interaction, whereby the membrane of one cell is attached to the membrane of the next cell, particularly in multicellular organisms. And you find that many cells make tissues, and many tissues make organs. So these cells that make up a tissue, all of them should work in concert, meaning they should work together. Therefore, for them to communicate from one cell to another, that contact between the membranes is what now helps them to transfer information from one cell to the other so that they can act together. For example, in muscle contraction, there is usually a wave of information flowing so that the cells can contract, causing all the muscles to contract, or the muscle tissue to contract as a unit. So that interaction comes through the membrane. And then some membranes are involved in energy transduction, like in the mitochondria and in the chloroplast. I had mentioned this when I was talking about mitochondria, that the process of cellular respiration or the process of photosynthesis happens on membranes. It happens using proteins which are embedded in particular membranes found in the mitochondria and in the cytoplasm. So those are the various functions of not particularly the cell membrane, but membranes. They can perform all these different functions. That's why it's a special organ. Now, the structure of the membrane has been studied for a while, but the structure that is normally used, known as the fluid mosaic model, was first proposed by Nicholson and Singer in 1972. This is what is normally the accepted model. We are going to see how that structure looks like. And what it says is that cell membrane is made up of two components. It's made up of mostly of lipids and proteins. We also have other substances like carbohydrates, but which are not in very large amounts. So we normally say the plasma membrane is a lipid bilayer, meaning it is made of two layers, okay? And don't confuse with the envelope I talked about, that one membrane is made of two layers, okay? So those lipid layer is made up of lipids known as phospholipids, and in those phospholipids are embedded the proteins. Now, a phospholipid is a lipid which is made up of lipids, which come from fatty acids, and a phosphate group. If you study again about biochemical compounds, you'll learn about functional groups, and one of them is a phosphate group, which now combines with the lipids to create what we call phospholipids. Now, phospholipids have what you call a hydrophilic head and a hydrophobic tail. If you study again biochemical molecules, you'll find that there are certain functional groups which are not polar, they are non-polar, therefore they are hydrophobic. Then there are certain sections which are polar, meaning they can interact with water, they are hydrophilic. So within this same phospholipid, there are certain regions which can interact with water and certain regions which we cannot interact with water. So we normally say this compound is amphipathic, meaning it has both hydrophilic and hydrophobic regions. And then the proteins that are attached to this membrane are either extrinsic on the outside, on the surface of the membrane, or intrinsic, meaning they travel across the membrane. I think let's look at it so that we understand what we mean by this. If I start with the proteins, you can see this protein molecule. This is the membrane. Okay, sorry, this is the membrane first. First of all, we said it is a double layer, a bilayer. There is this top layer and then this bottom layer. You can see these structures. So the proteins, like I said, one is traveling across and one is just found on the surface. I think here we don't have a diagram, but some of the protein do not travel all through. They are just found on the surface of the membrane. And we said the membrane has phospholipids. It's a phospholipid bilayer, so this layer, and each phospholipid has a hydrophilic head and a hydrophobic tail. So we say they have a head and a tail which looks something like this. If you study the structures of these particular molecules, you'll be able to see what we mean by the head and the tail. Now this is what we call the fluid mosaic model that was proposed by the scientists that I mentioned earlier. So the lipid bilayer, the phospholipids, act as structural background, while the membrane proteins contribute to the specialization of the cell, meaning they control the special activities of the cell. Therefore, the ratio of lipids to protein varies depending on the type of membrane, the type of cell, and the type of organism. So what we are saying there is that the cell membrane is made up of lipids and proteins. And the ratio of the proteins and the lipids would depend on what the cell is doing, or in which organism it is in, or which type of cell it is, or even which membrane. Is it the membrane that is found in the mitochondria? Is it the membrane that is found in the nucleus? And so forth. For example, the inner mitochondrial membrane, which I talked about, contains proteins which are involved in, they act as electron carriers. Therefore, they participate in transferring these electrons to create energy, which is stored in the form of ATP. Therefore, the inner mitochondrial membrane would have more proteins than lipids. Another membrane, the myelin sheath, which is found around the nerve cells, performs the function of insulation. Okay? So this is still a membrane, but it's performing a different function of insulation. Therefore, to insulate, you'd require more lipids than proteins, because proteins are able to conduct the kind of electricity you find in the nerve. Actually, you can see proteins have charges. Some proteins would have some charge, and that would help in conducting some of the energy, or in quotes, electricity that is traveling in the nerve cells. Therefore, they would not act as good insulators. So as you can see, these two examples, here you have more proteins, here you have more lipids, and both of them are membranes. Therefore, they would have different components. So, like we said, membrane lipids are amphipathic, meaning they are both hydrophilic and hydrophobic. And the components, as we said, they're made of phospholipids, but there are three main types of lipids that make up the membrane. There's the phosphoglycerides, which are phospholipids which have a glycerol background. Again, for you to understand this better, look at some structures of these phosphoglycerides to be able to see the glycerol structure, which forms the backbone of this particular lipid. We have sphingolipids, whose backbone is made of sphingosine, again, which is a structure, again, which I could not explain in words, but if you look at the structure in a textbook, you'll be able to see the difference. Again, cholesterol is made from a steroid or a sterol, and it has a very different structure from these other lipids. And it's in fact 50% of certain animal cells is made of cholesterol within the structure. And this is why we are sometimes told cholesterol is not good in high amounts in our nutrition, although the body already synthesizes cholesterol. If you have this in high amounts, it will end up in the membranes, and one work that cholesterol do in the membrane, they provide rigidity to the membrane. And therefore, if you have too much of it, the membrane becomes very rigid, and if now there are your veins and arteries, especially arteries, then they become too rigid and they are not able to move, and at some point might block the arteries and cause a problem, what we call atherosclerosis. So these different components in different amounts make up different types of membranes, depending on which role the membrane is playing. We also have membrane carbohydrates. Mostly they face either on the outside or on the inside. If you remember the diagram I've shown you, you will see them attached to lipids or attached to proteins on the outside of the membrane. When they're attached to proteins, they are known as glycoproteins. Glyco meaning the carbohydrate part, protein, the protein part, and 90% are linked to proteins. Then they can also be linked to lipids to form glycolipids. So most of the time you'd find them in the, I believe, in the receptors that are found on the cell membrane. Cell membrane proteins are grouped into three classes. We have integral proteins, which now penetrate through the membrane, like I had shown you in the diagram. We have peripheral proteins, which are located entirely on the outside or associated with surface membrane proteins. And then we have proteins that are anchored on the lipids. Now, these other proteins are independent. These ones are anchored on the lipids, and they're located outside. So these different proteins perform different functions. So as you can see, this protein is transversing the membrane, the lipid bilayer. This is also transversing. These ones are now transmembrane. They are all transversing the membrane. Here, these ones are only on the surface, but they are independent, but they are not going through. These ones mostly would probably act as receptors, and also this, you find the protein is attached to the lipid, okay, on the surface. Also, these are, again, proteins attached to probably the lipid. So we have those different types of proteins attached to the membrane, some running through, some on the surface, some attached but entering into the membrane, some attached on the top. So what are the functions of some of these integral membrane proteins? One, they act as receptors, like I'd said, where they bind specific substances and communicate information into the cell. Some act as channels, you know, a way through for certain substances, which especially ions and solutes, which are needed by the cell. So they provide a channel. If I could go back a bit here, so they would provide a channel for substances to run through the particular membrane. Then some act as electron transfer agents, like in the mitochondria, where we mentioned that they're involved in cellular respiration, in the cytoplasm, in the chloroplast, where they're also involved in photosynthetic. They are also amphipathic, meaning they have both hydrophilic and hydrophobic regions within their structure. Now, the ones which are transmembrane, the part which is inside the lipid has some hydrophobic characteristics. Okay. Then peripheral membranes, which are the membranes on the outside, the ones located on the inside, that is on the cytosol or the cytosolic surface, help in forming kind of a skeleton, that structure, the net, a net or a network that now provides a stable structure on the lower side of the membrane. They also provide mechanical support, where they anchor integral membrane proteins. So they provide an anchor for the proteins. Some of them act as enzymes, some as specialized coats. We have certain membranes which are specialized. Like, for example, we talk about the myelin sheath. It would be specialized the membrane would be a specialized coat. Others act as factors that transmit signals within the membrane. So where substances move through the membrane in different ways. Like we said, the membrane is a barrier, but it is a semi-permeable barrier, allowing certain substances to go through while denying certain substances to go through the membrane or entering the cell. Or, most importantly also, it denies certain substances from going out of the cell, because if a substance is required by the cell for its function, then it would be improper for that substance to move out of the cell. So it is retained within the cell by the actions of the cell membrane. So small molecules can be transported across the membrane by simple ways. With one, we have diffusion through the lipid bilayer, meaning we have certain substances like water which can easily diffuse through the cell membrane. So they don't require any special requirements. They move in and out of the membrane without being restricted. So they move in and out through what you call simple diffusion. We also have simple diffusion, meaning they just travel through, but they travel through the protein-lined channels. Like I had shown you, there are the structures of proteins that are transversing the bilayer. So some substances are allowed to pass through there, like some of the solutes which are important in the nutrition of the cell. We have also others which travel through facilitated transport, facilitated diffusion, or also facilitated transport using proteins. There are certain proteins that attach to the substance that needs to be transported, and they are moved through the membrane into the cell. So the fact that they are being attached to that protein, now the protein acts as a carrier to carry them across. So those ones are what we call the protein transporters. Then we have also what you call active transport. Now active transport is where now these substances are pumped across based on, they go opposite from diffusion. Diffusion is movement from high concentration to low concentration. So when you're moving from low concentration to high concentration, or against what we call the concentration gradient, you may need some energy to push the substance across. So that is what we call active transport, which is done through what we call the protein pumps. The substance is being pumped across because naturally by diffusion, the substance will not be able to travel across the membrane. So these processes use energy, and they use the ATP energy which we talked about, which is created by the mitochondria. So as you can see, the diagram again about endocytosis and exocytosis. Movement of large molecules. The other one was movement of small molecules. Movement of large molecules cannot just happen through the membrane. They're the ones that use this system of using vesicles, which I had explained earlier, substances coming in and substances going out. Endocytosis, when we are taking solid substances, we sometimes call it phagocytosis. And when you're taking liquid or fluid substances, we call it pinocytosis. And the substances now are able to transfer through. So let's finally look at our topic, the last topic today, of cell division. We have looked at cell membrane, cell structure, and a history about cell biology. Now let's look at cell division. Remember, the cell theory states that cells are created from other cells, meaning a cell divides to create a new cell. So cells are always dividing to create new cells. The parent cells divides to create daughter cells through the process what you call cell division. And you find that cell division is similar in all, nearly all cells. Okay, the process happens in the same way. And it is a continuous process, but we normally, as biologists, are able to break it down into various steps so that we can show what happens in the different steps. So it has phases, and these phases are what we call the cell cycle. Now we call it a cell cycle because cells are constantly dividing. So at one point, a cell would be found at one phase of the cycle. So a cycle is something that goes round. So it doesn't, have a beginning or an end. You are constantly going round. So a cell is either growing or dividing. Or in some cases, we may find that certain cells are resting, okay? But that is in very, very few cells. So cell cycle is a complex series of repeated stages of cellular events that leads to division of the cell material equally into daughter cells. So it's a constant process of growth and dividing. And in the process, it is very meticulous to ensure that each cell that is created is exactly same as the cell that it came from, so that we don't have things like mutation. So the cell cycle involves two phases. There is the interphase. This is the non-dividing stage. This is when the cell is growing and it is preparing itself for division. And then we have the mitotic phase. This is now the phase where the cell is dividing into two. So some cells undergo a specialized resting period, which we call the GO phase. Now this cell is neither growing or neither is it dividing. So it is, we call it a GO phase or G0 phase, where the cell is dormant and performing its normal functions, but it is not undergoing the cell cycle. Okay, so what is the interphase? Like I said, cell cycle is interphase and mitotic phase, growth phase and dividing phase. So the interphase, this is the longest period in the cell cycle, and it is the period in between the cell dividing. So the cell grows, it divides. So then it grows some more and divides. So the period between when the cell divided and when the cell dividing again is what we are calling the interphase. It is a long period, and it is comprised of three phases. The first phase, which we call the gap 1 phase, is when the cell begins to double in size. And now you can see the importance of the cell membrane being able to open up so that the cell can grow. And the cellular content is divided, so is duplicated, meaning the cytoplasm, the cytoplasm contents, the organelles, are being made copies of, because the cell is about to divide into two, and each of the new two cells should have the same substances that the original cell had. So if you had 3 mitochondria, then the new cell should also have 3 mitochondria. Therefore, the 3 has to multiply into 6, so that when there's a division, 3 would go to one side, another 3 would go to the other side. And then also happen to the various other organelles. Then there is also the other phase of synthetic phase, or sometimes we call the S phase. This is the second phase, and in this phase is where the chromosomes are duplicated. Remember, each cell has a specific number of chromosomes. Therefore, when the cell is to divide, the daughter cells must have the equal number of chromosomes as the original cell, unless they are the gamete cells, which are now created by another process known as meiosis, which is also part of cell division. Therefore, the chromosomes are copied so that they are now become pairs. Like, for example, the human being has 23 pairs of chromosomes. So the 23 pairs, meaning 46 chromosomes, would be multiplied so that you'll end up with 96 chromosomes, or 46 pairs. So that when the cell divides, the new cell will have 23 pairs, another cell will have 23 pairs. So that happens in the synthetic phase. And then lastly, in the interphase, remember this is when the cell is just growing, it is not dividing. We have the gap 2 phase. This is now where the cell begins to assemble what we call cytoplasmic material, which are necessary for cell division. As you're going to see, some of the cytoplasmic material are things like the mitotic spindle, which will help in the division of the cell, and other substances which now help in the process of the cell division. So in the M phase, the mitotic phase, Now the cell is dividing. What is involved here? Two things. The nucleus will divide, and the process is known as karyokinesis, and then the cytoplasm will divide, and the process known as cytokinesis. We have two types of M phase. We have mitotic phase, in which when the cell divides, the daughter cells that are created, first of all, two daughter cells are created, and second, they have the exact same number of chromosomes as the parent cell. The process of mitosis is for cell division, and meaning for growth, is cell division for growth and for development, and also for things like repair or replacement of cells that are worn out. Meiosis is when we are doing gamete production. Now this process ends up with, the cell division ends up with four daughter cells, and each of those daughter cells has half the number of chromosomes as the parent cell. Again, that can be shown in the various steps involved. So this is a picture depicting the whole cell cycle. You have your G1 phase, where cellular contents, excluding the chromosomes, are duplicated. The S phase, where the chromosomes are duplicated. And then the G2 phase, where there's double checking of what has been done at these two stages. And then the repairs are done, and the cytoplasm begins to prepare for the process of division. And then mitosis or meiosis is now the process of the cell dividing. Cytokinesis, like I said, is the division of the cytoplasm. So mitosis mostly would be referring to the division of the nucleus. But mitosis is the entire process of dividing, but it leans more towards explaining the division that is happening in the nucleus and not the cytoplasm. So here you'll have karyokinesis, and then after that you have cytokinesis. And then there are certain cells that can go into rest away from the cell cycle, which you're calling the G0 phase. Now, what factors regulate the cell cycle? Regulation means the process will happen, but how fast or slow does it happen? So it is regulated, or meaning controlled. So cell-to-cell contact is very important. When cells touch each other, they help in communicating the need for cell division or the lack of it. The process, the disease, like cancer. Cancer now affects the DNA of the cell and causes the process of cell division to go out of control. Remember, I talked about the nucleus controlling the functions of the cell. So cancer can cause the nucleus to lose control, and the cell cycle now becomes unregulated and begins to happen uncontrollably. We have environmental factors like low temperature would affect cell division, lack of nutrients, and lack of hormones. Hormones are involved in giving directions for growth and cell division. Therefore, lack of hormones could cause lack of growth. Like, for example, we normally grow as a result of growth hormones. So when these growth hormones are high in our system, like when you're in the childhood, the growth is there. But when you grow older, the amount of hormones reduce, therefore less communications to cells to grow. So as we conclude, we have talked about interphase, which is growth. Now division, the division does not just happen. It happens in steps. There are 4 steps: prophase, metaphase, anaphase, and telophase. These are not, again, distinct steps, but they're just trying to explain the activities that happen during cell division. So the primary activity that happens during cell division is that the chromosomes are first, they've been doubled during interphase. So the main thing that happens during mitosis is that the chromosomes are aligned at a certain point so that they can now be separated into the 2 cells equally. Because if that doesn't happen correctly, the new cells will have the wrong genetic information or distorted genetic information, which will end up with creating distorted organisms. Therefore, that separation has to be very accurate. So the process of mitosis is trying to ensure that that division is very, very accurate. So the chromosomes are brought to one line, they are aligned correctly, and then they are divided. And that is what happens during mitosis. So what happens during prophase, the first step of mitosis? This is the longest phase. What happens is the nuclear membrane disintegrates and the nucleolus, so that membrane-bound nucleus disappears. And then a structure known as the centrosome creates the centrioles, which now migrate to different opposite parts of the cell. These centrioles would allow for production of microtubules to make what we call the mitotic spindle. Also during the process, the chromosomes condense, okay? They condense and appear as two identical chromatids attached together by a central structure known as the centromere. Then the chromosomes now migrate to the central part of the cell, which we are calling the metaphase plate, a central part as you're going to see in the diagram. Then the spindle fibers attached to the chromatids at the centromere through some protein structures known as the kinetochore. So if you look at this diagram, this is the cell before it is dividing. Then you can see the chromosomes are simply straight strands. When they multiply, they become two strands. This one strand running like this, another strand running like that. In between, they are attached together by the centromere. Another thing, the centrosome creates the centrioles, which now begin to migrate to opposite sides of the cell. And this is now what we call the prophase, the preparation for cell division. Then the next two steps, metaphase, the condensed chromosomes reach the cell equator, meaning the metaphase plates, the central part. Then we have anaphase, where the centromeres divide, the sister chromatids are pulled apart. They are pulled apart by the spindle fibers, which are attached to the kinetochore. And then half of the chromosomes move to one side of the cell, and another set moves to another side of the cell. Now, once that happens, as I'm going to show you in the diagram, we have now the final phase, which we call the telophase. In the final phase, the telophase, what happens? It is a reverse of the prophase, meaning the things that happened in the prophase are now reversed back. The nuclear membrane reforms around the set of chromosomes that have been separated to the different poles of the cell. The chromosomes uncondense, they lengthen, and then we cannot even see them with a microscope. The spindle fiber disappears, which was created again at the prophase, and then the nucleolus reforms and becomes visible again. So this is what happens. At metaphase, the chromosomes now are aligned at the central part of the cell, which we are calling the metaphase plate. And you can see they are attached to the spindle fiber like that, so that when you have anaphase here, the next step, the spindle fibers pull apart so that these two sister chromatids, this is chromatid 1, chromatid 2, they are separated from the centromere here. They are separated and pulled in opposite directions. And then finally, telophase, we said, is a reverse. Now things begin to reverse. The nucleolus reappears, the cell begins to pinch and separate into two, what we are calling cytokinesis, so that two cells are now being created. So what is the significance of mitosis? Equal distribution of chromosomes, so that the parent cell is similar to the daughter cell. That the chromosomes in the nucleus of the parent cell are equal to the chromosomes in the nucleus of the daughter cell. It is a method in which body cells, also called somatic cells, are able to multiply. So when the body is growing, the cells are simply dividing, they use the process of mitosis. Asexual reproduction, especially in plants, When we do what we call vegetative propagation, where you take a piece of the plant and grow it and plant it so that it grows into a new plant, usually the new plant is created from existing cells. And that is through the process of mitosis. The cells are dividing and creating the new plant. Then we have, it is a mode of reproduction in unicellular organisms. Things like bacteria, they do not have sexual reproduction. So what they use is the mitosis process to create new organisms. Cell replacement, like for example, the skin is constantly being replaced. Skin cells are wearing out, new skin cells are created. So the process of mitosis creates new cells to replace the old ones. And also regeneration of some body parts. We have certain body parts like the liver, which can be regenerated from existing cells. We have certain organisms like a lizard. I'm sure as a young boy or girl, you cut the tail of a lizard, and after a while you saw the tail growing back. That is regeneration. And that regeneration is made possible by cell division or mitosis. Now, to conclude our class, I would want to give you an assignment. Read more on meiosis, which is the other process that we have during cell division. Now, meiosis creates the gamete cells, which can now join together later to create a zygote. Meiosis, like I said, is different from mitosis, whereby there are two divisions. So I'd want you to check those two divisions and see how they correlate with mitosis and how we end up with half the number of chromosomes compared to mitosis, where the parent and the daughter cell have equal amounts of chromosomes. So with that, class, I'd want to conclude the lecture, and thank you for attending.
We are in a digital era, and Mount Kenya University knows this. The following are the steps to follow so as to complete your online application. Download the application form from the website www.mku.ac.ke. Attach copies of your academic certificates and ID. Pay the application fees via M-Pesa pay bill number 270988. Your ID is the account number. 2,000 shillings is the charge for a postgraduate. You can also deposit in the bank accounts provided on the website. Then email all the above to apply@mku.ac.ke.